Next, we're going to be looking at Chapter 20, Part C. We're focusing on the cellular immune arm of the adaptive defense system, which focuses on T cells and how they are able to provide defense against intracellular antigens. An example of an intracellular antigen includes viruses, bacteria, cancerous cells, abnormal cells, even foreign or transplanted cells. Some T cells directly kill cells, whereas others release chemicals that regulate the immune response. T cells are more complex than B cells, both in classification and function. There are two populations of T cells based on which cell differentiation glycoprotein receptors are displayed on their surface. CD4 cells usually become helper T cells, which activate B cells, other T cells, and macrophages. They essentially direct the adaptive immune response. CD4 cells can also become regulatory T cells, which moderate the immune response. CD4 cells are also able to become memory T cells. Even CD8 cells can become memory T cells, but CD8 cells also become cytotoxic T cells that are capable of destroying cells harboring foreign antigens. Helper, cytotoxic, and regulatory T cells are activated T cells. The naive T cells are simply termed CD4 and CD8. Once formed in the bone marrow, T progenitor cells migrate to the thymus, hence the name T cell, and they migrate to the thymus to mature and become activated T cells. While in the thymus, the developing T cells start to express T cell receptors and other receptors called CD4 and CD8 receptors. Those are the glycoproteins we previously talked about. All T cells express T cell receptors, and it's either a CD4 or CD8 glycoprotein that's also expressed on the T cell, but not both. So some T cells will express CD4 and others will express CD8. That's what we see in this image with CD4 being shown on the left and CD8 being shown on the right. Unlike antibodies, which can bind to antigens directly, T cell receptors can only recognize antigens that are bound to certain receptor molecules. These receptor molecules are called major histocompatibility complexes. There is a class 1 and class 2 MHC protein. These MHC molecules are membrane-bound surface receptors on antigen-presenting cells, like dendritic cells and macrophages. CD4 and CD8 cells play a role in T cell recognition and activation by binding to either MHC class 1 or MHC class 2. MHC class 2 is only found on antigen-presenting cells, whereas MHC class 1 is found on all cells that have a nucleus, so they are not found on red blood cells, which are anucleate. T cells respond only to those processed fragments of antigens that are displayed on the surface of cells by those MHC proteins, or major histocompatibility complexes. The antigen presentation is vital for the activation of naive T cells and normal functioning of effector T cells. So let's look closer at those two classes, class 1 and class 2 MHC proteins. Again, class 1 is displayed by all cells except red blood cells, and class 2 is displayed by antigen-presenting cells. The antigen-presenting cells, remember, are the dendritic cells, macrophages, and B cells. Both types are synthesized in the endoplasmic reticulum and bind to peptide fragments. These MHC proteins are what would contain either a self or non-self antigen. If we're looking at the class 1 MHC protein, we want to note that it binds with short fragment, which is an 8 to 9 amino acid length endogenous antigen. This protein is synthesized inside the cell. An endogenous antigen could be a self antigen or a non-self antigen. A normal protein of the cell would be a self antigen. An abnormal protein found in infected or abnormal cells would be a non-self antigen. The class 1 MHC is crucial for CD8 cell activation because it acts as an antigen holder. It forms the self part, essentially, that T cells must recognize. So it informs the cytotoxic T cell of microorganisms hiding in cells. Whereas the class 2 MHC proteins bind with longer fragments, so 14 to 17 amino acids long, usually of exogenous, which is extracellular antigens, that have been engulfed and broken down in a phagolysosome by an antigen-presenting cell. The class 2 MHC proteins are recognized by T helper cells, so they're able to signal CD4 cells that help is required. So if we look at the role of MHC protein in cellular immunity, we can see that the class 1 MHC proteins are displayed by all nucleated cells, including the antigen-presenting cells, but class 2 MHC proteins are only located on dendritic cells, macrophages, and B cells, which are the antigen-presenting cells. The MHC proteins are essentially a protein chair, which would hold a self or non-self antigen. MHC1 proteins are recognized by naive CD8 cells and cytotoxic T cells, whereas the class 2 MHC proteins are recognized by naive CD4 cells and the helper T cells. MHC class 1 proteins are looking at or for endogenous pathogens or proteins, which could be made by cancerous cells. So that's the type of protein that you would have on an MHC class 1 protein, whereas in the MHC class 2 proteins, we'd be looking at exogenous or phagocytosed extracellular pathogens sitting in the divot of the MHC protein chair. Cells that display foreign antigens on MHC send this message if the foreign antigen is displayed on class 1 MHC. So here we have an example. If the cell's an antigen-presenting cell and it comes into contact with an MHC class 1 protein displaying a foreign antigen, it would say, it would think that the cell is saying, I belong to self, but have captured a foreign invader. And this is what it looks like. So kill any cell that displays it. So what that's saying is if an antigen-presenting cell displays a foreign antigen on a class 1 MHC, it is saying, I caught a foreign thing, please kill anything that looks like it. Whereas if the cell is just a normal cell, but it's displaying a non-self antigen, that normal cell is going to say, I belong here, but something's happened. I've been invaded. Maybe I became cancerous. And that's why I'm presenting an abnormal non-self antigen instead of my normal self antigen. So please kill me because an infected cell can't be saved. Whereas with our class 2 MHC proteins, if we have a foreign antigen being presented in that class 2 MHC protein and it's located on an antigen-presenting cell, what that antigen-presenting cell is saying is, I'm supposed to be here. I'm supposed to be in the human body, but I've captured again a foreign invader. I know what it looks like. Help me mount a defense against it. So generally, we find that the class 2 MHC proteins, when displayed, because they're only displayed on the antigen-presenting cells, present to T cells. They talk to T helper cells, and they're going to ask for help in mounting an immune response. Whereas the antigen-presenting cells, when they present to T cytotoxic cells, they're going to present on MHC1, and they're not asking for help. They're asking for that T cytotoxic cell to directly kill anything that looks like this foreign antigen. We also need to talk about MHC restriction, which is talking about CD4 and CD8 cells and the fact that they have different requirements for MHC proteins in order to present antigens. The CD4 cells that become T helper cells are restricted to binding to only class 2 MHC proteins, typically on the antigen-presenting cell surfaces, which we just saw in that last diagram, whereas the CD8 cells that become cytotoxic T cells are restricted to binding only class 1 MHC proteins found on every cell surface. That even includes the antigen-presenting surfaces. Once activated, the cytotoxic T cells would seek out the same antigen on a class 1 MHC protein on any cell. The dilemma here is when antigen-presenting cells need to be enabled by CD8 cells to be activated by non-endogenous phagocytosed proteins. The dendritic cells are actually the ones that are able to get around this. Another reason why we call the dendritic cells the best antigen-presenting cells. They're able to get around this problem by engulfing dying virus-infected or tumor cells, then displaying engulfed cells, endogenous proteins on both class 1 and class 2 MHCs. The dendrites of dendritic cells can also import antigens via temporary gap junctions with infected cells rather than engulf them. So it's the dendritic cells that are able to solve the dilemma for antigen-presenting cells, which is how to enable CD8 cells to be activated by non-endogenous phagocytosed proteins. T cells can be activated only when antigen is presented to them, which makes sense. We don't want them to just be activated suddenly because some of them can directly kill cells. Activation is a two-step process, therefore. It is going to consist of antigen binding to a T cell receptor, but also a co-stimulation, which is sometimes called the second handshake. Both steps are going to occur on the surface of an antigen presenting cell, or at the surface of an antigen presenting cell, and both are required for the clonal selection of the T cell. So before we get proliferation and differentiation, we have to clonally select this T cell. So step one is when the antigen binds. So an antigen would bind to a T cell receptor. This antigen must be displayed on the correct major histocompatibility complex, or MHC protein. The MHC and antigen complex would be located on the antigen presenting cell, and that complex would be what binds to the T cell receptor. The T cell receptor must perform a double recognition. It has to not only recognize MHC, but also the foreign antigen it displays. And then when the T cell receptor binds to the MHC antigen complex, if those two recognitions or double recognition is made, that would trigger multiple intracellular signaling pathways that would start T cell activation. Other T cell surface proteins that would be involved in this process include CD4 and CD8, depending on the type of T cell we're talking about. Those are the glycoproteins that are found on naive T lymphocytes. So we want to help maintain coupling during antigen recognition of these glycoproteins to the surface of the antigen presenting cell. Step two, the co-stimulation, is going to be required for the complete T cell activation. It would require the T cell to also bind to one or more co-stimulatory signals. A common co-stimulatory signal is a B7 protein. The co-stimulatory signals are generally located on the antigen presenting cell. Without the co-stimulation, then anergy, which is essentially shutting down of the T cell, that would occur. It would cause the anergic T cell to become tolerant to that antigen, unable to divide, and would not cause it to secrete cytokines. So this two-step process is required, but it's a safeguard against the unwanted activation of T cells. Once we activate a T cell, so we have clonal selection, proliferation, and differentiation can occur. This is where T cells are not only activated, but enlarge and proliferate in response to the secretion of cytokines. The differentiation of T cells is in order for them to perform functions according to their T cell class. So that would be either T regulatory cells, T cytotoxic cells, or T helper cells. Primary T cell responses peak within a week. T cell apoptosis would occur between about day 7 and 30 after the initial peak. Activated T cells are a hazard because they produce large amounts of inflammatory cytokines, so we'd want to eventually shut this off. And that inflammatory cytokine production, if it goes unchecked, could result in hyperplasia or cancer if it's not cleared from the system. Memory T cells, however, would remain and mediate a secondary immune response. So in figure 20.17, we're just looking at clonal selection of T cells and how that involves not only the simultaneous recognition of self, which is MHC, but non-self, which is the foreign antigen. So antigen presentation, here we're seeing a dendritic cell, which has engulfed an exogenous antigen, processed it, and displayed that fragment on a class 2 MHC protein. We see that it also has a co-stimulatory molecule, and it's going to be able to combine next to the T cell receptor. This is a part of the double recognition, which not only requires the CD4 T cell to recognize the antigen MHC complex, but it requires a TCR, which is the T cell receptor, and the CD4 glycoprotein on the T cell to bind to the antigen MHC complex. Depending on the type of T cell we're looking at, it might not be a CD4 protein, it could be a CD8, but we're looking here at a naive CD4 cell that when activated will become a helper T cell. The co-stimulatory molecule again must also bind, that's usually a B7 protein. If all of this occurs, that double recognition, that co-stimulation, we get clone selection and clone formation, which will then lead us into proliferation and differentiation. Here we have the activated CD4 cells proliferate, and they're becoming memory and effector cells. The CD4 cells, when activated, become helper T cells as their effector cell. But don't forget, both helper and cytotoxic cells can generate memory T cells. We also want to mention, what are cytokines? We mentioned them briefly when we talked about the innate system and when we started getting into things like interferon and complement, but we've not gone into a lot of detail. Cytokines are chemical messengers of the immune system that mediate cell development, differentiation, and responses in the immune system. Basically, think of them as the adaptive defense chemicals. They do include interferons and interleukins. Interleukin 1 and interleukin 2 are common cytokines, and interleukin 1 is released by macrophages and stimulates T cells to release interleukin 2. So it's a little bit of a cascade pathway. And the release of interleukin 2 would synthesize more interleukin 2 receptors. So we just want to amplify the ability of interleukin 2, which is a key growth factor, to bind to other T cells because it acts on the same cells that release it. This encourages activated T cells to divide rapidly, so it's a proliferation and differentiation chemical. Other cytokines are also able to amplify and regulate the innate and adaptive responses. An example would be gamma interferon, which is a type of cytokine that enhances the killing power of macrophages. Here is just an overview of selected cytokines. So we're looking, for example, at alpha and beta and gamma interferon, which we've talked about before. Cytokine is just a general term for adaptive immune system chemical. We even have a list here of interleukins. Be familiar with interleukin 1 and 2, and then take a quick look at interleukin 4. And then there's things like TGF-beta and tumor necrosis factors. They will come up, but they will not be a main focus for Chapter 20. Next, we're going to look specifically at the roles of the effector T cells. So we'll be looking at the T helper cells, T regulatory cells, and T cytotoxic cells. First, let's look at the helper T cell, which plays a central role in the adaptive immune response and essentially able to direct both the humoral and cellular arms, because once primed by an antigen-presenting cell, by the presentation of a foreign antigen, the helper T cell will be able to help activate B cells, other T cells, induce T and B cell proliferation, and secrete cytokines that recruit other immune cells. So essentially, the helper T cell is the director of the adaptive defense. So without the T helper cell, there's no immune response. The activation of B cells is going to be aided by T helper cells. Helper T cells interact directly with B cells, displaying antigen fragments bound to MHC2 receptors. This stimulates B cells to divide more rapidly and even begin antibody formation. The B cells may be activated without T helper cells by binding to T cell independent antigens, but most antigens are actually T cell dependent antigens and require the T helper co-stimulation in order to activate B cells and cause their proliferation and differentiation. So here in figure 20.18, we're looking at the role of helper T cells in mobilizing the humoral and cellular immunity response. In part A, we're looking at its role in humoral immunity. And the role in humoral immunity is the activation of the B cell. Here we have a B cell displaying its processed antigen, and it's displaying that to a T helper cell. We have a CD4 protein that is co-stimulating this B cell receptor. And by talking to the T helper, the B cell is getting the release of interleukin 4 and other cytokines. So the release of interleukin 4 and other cytokines by the T helper cell is going to be co-stimulatory in the fact that it's not only going to activate the B cell, but lead to proliferation and differentiation of this T cell. Activation of CD8 cells requires the T helper cell as well, because to become activated, the CD8 cells require talking to the T helper cells. CD8 cells are naive cells that become cytotoxic T cells when activated. We know that T helper cells cause dendritic cells to express co-stimulatory molecules, the ones that we need for CD8 cell activation. So that's what we're seeing here is the helper T cell helping in cellular immunity. So first and foremost, the T helper cell has to bind to the dendritic cell. It's binding to class 2 MHC protein displaying a foreign antigen. And we're going to see that the CD4 protein is bound to that MHC2 protein. That causes the release of interleukin 2 from the T helper cell. And because the dendritic cell is bound also to the CD8 cell via both the CD8 protein and a co-stimulatory molecule, the interleukin-2 is going to be close enough when released to act on the CD8 cell. And the interleukin-2 will be able to help activate the CD8 cell. The binding of the dendritic cell and the CD8 cell is via an MHC class 1 protein, and that MHC class 1 protein is also displaying a foreign antigen. The roles of specific effector T cells include the amplification of innate defenses. So we want to see the amplification of not only the activation of macrophages, which lead to more potent phagocytic killers, but also the amplification of responses of the innate immune system, like inflammation, and the mobilization of lymphocytes and macrophages that attract other types of white blood cells. So we want to increase leukocytosis or phagocyte mobilization. There's also subsets of T helper cells like T helper 1 and 2 and 17, and they do play a role in autoimmune diseases or promoting allergy or defending against parasitic worms or mobilizing eosinophils, but it's the T helper 1 that mediates most aspect of cellular immunity. Let's also look at cytotoxic T cells, which directly attack and kill other cells. So it is important to note that cytotoxic T cells are direct in their action. The activated cytotoxic T cells actually circulate in the blood and lymph or the body's humors. We can also find some in lymphoid organs like lymph nodes. They're able to search body cells that display foreign antigens they recognize on the correct MHC protein. Activated cytotoxic T cells are going to target virus-infected cells, cells with intracellular bacteria or parasites, cancerous cells, or foreign cells from transfusions or transplants. So they're looking for endogenous antigens. Cytotoxic T cells deliver a lethal hit using two mechanisms. This mechanism sees the release of perforins and granzymes by exocytosis. We also see that the T cytotoxic cell binds a specific membrane receptor on the target cell in order to help stimulate this apoptosis. The perforins create pores through which granzymes enter the target cell, whereas the granzymes stimulate apoptosis. Both cytotoxic T cells and NK cells use perforins and granzymes as a mechanism of action for killing infected cells. And that's what we're seeing here in figure 20.19a is a cytotoxic T cell attack an infected or cancerous cell. It first identifies the foreign antigen on an MHC1 protein, then binds tightly to it. Once it binds tightly to it, it releases perforin and granzyme molecules from its granules by exocytosis. The perforin molecules insert into the target cell membrane. They polymerize and form a transmembrane pore, and then the granzymes enter into the infected cell through the pore. The granzymes are going to, once inside, activate enzymes that trigger apoptosis. And then the T cell, the cytotoxic T cell, detaches and then searches for another prey. Natural killer cells are also able to recognize other signs of abnormality that cytotoxic T cells do not look for. Remember, natural killer cells are non-phagocytic, but they are similar to cytotoxic T cells. But natural killer cells can actually recognize a lack of MHC class 1, whereas cytotoxic T cells are looking for MHC1 proteins holding foreign molecules. Natural killer cells can also recognize antibodies coating a target cell, as well as different surface markers seen on stress cells or abnormal cells. So NK cells are, while they're able to use the same mechanisms as cytotoxic T cells for killing their target cells, their immune surveillance is slightly different. The NK and TC cells, or cytotoxic T cells, still both prowl the body looking for markers that they each recognize. And of course, we have to mention the regulatory T cells, or Treg cells, which dampen an immune response by direct contact or by secreting inhibitory cytokines, such as interleukin-10 and TGF-beta. And TGF-beta stands for transforming growth factor beta. They are both important in preventing autoimmune reactions. And so we would see that T regulatory cells are able to suppress self-reactive lymphocytes in the periphery, which is in tissues outside of lymphoid organs. And we are looking into research using them to induce the tolerance of transplanted tissue because they're able to, again, suppress self-reactive lymphocytes. And then here, table 20.8 is just looking at the cells and molecules of the adaptive immune response. Another good summary table looking not only at the effector cells of the humoral and cell-mediated immune response, but looking at the molecules that help therein. And then one of our last topics is going to be organ transplant and the prevention of rejection. So the most common type of organ transplant is an allograft, which is a transplant from the same species. Success does depend on the similarity of tissues. We would test ABO blood type, other blood antigens, MHC antigens, try to test all of those types of antigens to make sure that we have a great match. After surgery, the patient would then be treated with immunosuppressive therapy. And many of these therapies have severe side effects because when you suppress a patient's immune system, that means that they're not able to protect their body from foreign agents, such as simple bacterial and viral infections that they normally would be able to fight off. That could lead to death among transplant recipients if they get an opportunistic infection. So under the best circumstances, rejection is expected after 10 years in about 50% of patients. Other things we want to mention here are immunodeficiencies, which are just congenital or acquired conditions that impair function or production of immune cells or molecules. One such immunodeficiency is severe combined immunodeficiency syndrome, abbreviated SCID. This is sometimes called the bubble boy disease, but it's a genetic defect that causes a marked deficit in the amount of B and T cells. A defective deaminase enzyme, adenosine deaminase, or ADA enzyme, is what allows for the accumulation of metabolites that become lethal to certain T cells, sometimes lethal to T helper cells. So this can be fatal if it's untreated, and the treatment is bone marrow transplants. There's also Hodgkin's disease, which is an acquired immunodeficiency that causes cancer of B cells. So Hodgkin's disease, sometimes called Hodgkin's lymphoma, is essentially a cancer of B cells which depresses the lymph node cells and thus leads to the immunodeficiency or lack of active B cells. There's also the acquired immune deficiency syndrome or AIDS, and the human immunodeficiency virus cripples the immune system by interfering with activity of helper T cells. And this is characterized by severe weight loss, night sweats, swollen lymph nodes, and opportunistic infections, including pneumocystis pneumonia and Kaposi sarcoma. HIV is essentially transmitted via bodily fluids, so blood, semen, and vaginal secretions. HIV enters the body via blood transfusions, blood-contaminated needles, sexual intercourse, and oral sex, or from mother to fetus. The reason that HIV is so dangerous is because it destroys T helper cells, which direct the adaptive arm of the immune response. So that suppresses pretty much the entire side of the adaptive system. And so it definitely reduces the ability of the human body to fight off infection. HIV multiplies in lymph nodes throughout the asymptomatic period inside the T helper cells. An individual can go 10 years asymptomatic if untreated, and symptoms generally begin when the immune system collapses after that asymptomatic period. The virus can also invade the brain, which can lead to dementia. HIV is a coded glycoprotein complex that attaches to the CD4 receptors on naive CD4 cells. That allows HIV to enter the cell and use reverse transcriptase to produce DNA from its own viral RNA. So it's a reverse transcriptase virus. HIV reverse transcriptase is prone to frequent errors. That makes it really hard to fight the HIV virus because of the high mutation rate and resistance to drugs. The DNA copy is called a provirus, and it directs the host cell to make viral RNA and proteins, and that enables the virus to reproduce within the T helper cells. Currently, there is no cure for AIDS that has been found, but there are 4 major classes of antivirals that are used in combination and have helped to reduce the virus or viral titer of active virus inside an individual. Another topic we want to look at is autoimmune diseases. Autoimmune disease result when the immune system loses the ability to distinguish self from foreign. Autoimmunity production of antibodies, called autoantibodies, are oftentimes going to sensitize cytotoxic T cells and destroy body tissues. Examples of autoimmune diseases include rheumatoid arthritis, myasthenia gravis, multiple sclerosis, Graves' disease, type 1 diabetes mellitus, systemic lupus erythematosus, which affects multiple organs, and glomerulonephritis, which damages the kidneys. Essentially, what an autoimmune disease is a disease where the body attacks itself. For treatment of autoimmune diseases, we need to suppress the immune system because we need to suppress the immune system from attacking your own body. So anti-inflammatory drugs, drugs that block cytokine action or block that co-stimulatory molecule on T cells, those can all be used to suppress the entire immune system. But there is research into using activated regulatory T cells in order to reintroduce self-tolerance. This might be reintroduced using vaccines, which direct antibodies against self-reactive immune cells. Because failure of self-tolerance causes this autoimmunity condition, and it could be caused by, we think, weakly self-reactive lymphocytes that are activated when they bind to body proteins, and these foreign antigens that resemble self-antigens begin to kind of cross-reacting with these body proteins. And the appearance of new self-antigens because of these weak self-reactive lymphocytes binding to body proteins is generated, we think, by gene mutations, changes in self-antigens by hapten attachment or infectious damage, and release of novel self-antigens by trauma to a barrier. And then we still want to mention hypersensitivities, which are immune responses to perceived, otherwise harmless threats that cause tissue damage. So kind of like allergies or allergic reactions. There's different types of hypersensitivities based on their time course and whether or not they use antibodies or T cells. Antibodies are just going to cause immediate or subacute hypersensitivities, whereas T cells cause delayed hypersensitivities. For example, an immediate hypersensitivity would be allergies, and it usually begins in seconds after contact with the allergen and the antigen that causes the allergic reaction. The initial contact with the allergen tends to be asymptomatic, and it basically is a sensitizing stage. Activated IgE is going to be produced during this asymptomatic sensitizing stage, and it's produced against the antigen that binds to mast cells and basophils. And so a later or secondary encounter with the same allergen wouldn't be asymptomatic. The secondary encounter actually causes a flood of histamine released from the IgEs, and that would result in an induced inflammatory response or allergies. The histamines would cause vasodilation, increased vascular permeability. It would lead to symptoms like a runny nose, itchy and watery eyes, hives, as well as asthma, which can occur if the allergen is inhaled. Antihistamines are usually needed for control of immediate hypersensitivities. Allergic reactions can definitely be local or systemic. Local reactions involve mast cells of the skin or respiratory or gastrointestinal mucosa. A systemic immediate hypersensitivity is very dangerous. An example would be anaphylactic shock, for example, from a bee sting, because it's usually seen with injected allergens. It would cause the bronchioles to constrict, making breathing difficult, as well as vasodilation that would result not only in low blood volume, but would cause circulatory collapse. The treatment would be the immediate administration of epinephrine. Here in figure 20.20, we're looking at the mechanism of an acute allergic reaction, which is an example of an immediate hypersensitivity. So in step 1, we would have the sensitization step wherein an allergen would come into contact with a plasma cell, which would produce antibodies. Those antibodies tend to be IgE, and those IgE antibodies would attach themselves to a mast cell. Then the next time that someone comes into contact with that allergen, it wouldn't cause the production of antibodies. Instead, that antigen would bind to the antibodies that have attached themselves to this particular mast cell or basophil. And when they bind to that mast cell or basophil, that would cause a release of histamine. The release of histamine would cause vasodilation and increase vascular permeability, as well as things like the production of mucus and the contraction of smooth muscle. This could cause an outpouring of fluid from capillaries, the release of mucus, or constriction of smooth muscle of the respiratory passages, which could cause the bronchioles to constrict. All very dangerous, especially if it becomes a systemic allergic reaction. A subacute hypersensitivity would be caused by IgM and IgG when they are transferred via the blood plasma or serum. Much slower onset than our acute hypersensitivities. It does have a slightly longer duration, however, about 10 to 15 hours. Some of these subacute hypersensitivities are termed type 2 cytotoxic reactions, wherein antibodies bind to antigens on specific body cells and stimulate phagocytosis and complement-mediated lysis of cellular antigens. An example of a cytotoxic reaction is a mismatched blood transfusion reaction, wherein the body would not only agglutinate the mismatched blood, but then try to lyse the blood cells. Another example of a subacute hypersensitivity would be an immune complex type 2 hypersensitivity, wherein antigens widely distributed in the body or blood would form antigen-antibody complexes that are insoluble. These complexes would not be able to be cleared from a particular area of the body, and therefore cause intense inflammation, local cell lysis, and cell killing by neutrophils. An example of an immune complex type 2 hypersensitivity is systemic lupus erythematosus, or SLE. And then you have delayed hypersensitivities, which have an even slower onset of 1 to 3 days. This would be a type 4 delayed hypersensitivity, wherein the mechanism would depend on helper T cells, and cytokine-activated macrophages and cytotoxic T cells would be what causes damage. An example would be allergic contact dermatitis or coming in contact with something like poison ivy. The agents are essentially acting as haptens and must combine with proteins of the body in order to be immunogenic and reactive. A TB skin test is actually an example of a test that depends on this delayed hypersensitivity reaction. And that's it for type C. Make sure to go back and review the most MHC type 1, MHC type 2, and where they're located and how they act in comparison to the naive CD8 and CD4 cells.